Hello, my name is Marcus Doyle. I'm a master's student in the TCN lab here at Western, supervised by Dr. Tim Bussey and Dr. Lisa Succedo. So I want to discuss with you my master's thesis project today, which focuses on the neural circuitry underlying sustained attention. So why sustained attention? Well, sustained attention is crucial for the activities of daily living, and it's disturbed in certain neuropsychiatric diseases such as schizophrenia. So we know that parvalbumin interneurons play a crucial role in sustained attention. This work has been supported by numerous studies, but in particular, I wanted to note the PhD student's work in my lab, known as Tyler Dexter. Additionally, the cholinergic system, the prefrontal cortex, is also known to play a crucial role in sustained attention. So we know that parvalbumin interneurons in the prefrontal cortex express both muscarinic and nicotinic cholinergic receptors postsynaptically. So the question remains, does cholinergic modulation of parvalbumin interneurons play a crucial role or a necessary role in sustained attention? So to address this question, my project takes on two parts. In both, the sustained attention task, known as the rodent continuous performance task, which utilizes touchscreen technology, will be used. So essentially, in a hit condition, a mouse learns to respond to the S-positive stimulus in order to receive a milkshake food reward, while in an S- or non-target stimuli condition, the animal learns to non-respond. Their performance on this task is indicative of sustained attention. So what my two experiments plan to do is to, the first experiment plans to indirectly look at parvalbumin activity in the prefrontal cortex by analyzing the calcium activity or calcium flow of parvalbumin interneurons in the prefrontal cortex of these mice, while at the same time applying a cholinergic antagonist to the prefrontal cortex and measuring their performance on the sustained attention task I mentioned earlier. So we wanted to see whether these cholinergic inactivation would lead to changes in parvalbumin calcium dynamics, indirectly possibly relating to their activity levels. Following this, we wanted to combine optogenetics, specifically inhibitory optogenetics, with the sustained attention task to see whether the cholinergic system plays a necessary role in sustained attention. We chose to use optogenetics in this example because it's better able to temporarily target the parts of the task where attention is most utilized, allowing us to isolate out specifically the attention portions of the task. So in order to do this, we infected mice with an inhibitory opsin known as halorodopsin, and we then shine yellow light specific for halorodopsin to activate these inhibitory opsins, leading to the inactivation of Chad-expressing cholinergic neurons in the prefrontal cortex, and therefore the inactivation of the cholinergic system in the prefrontal cortex. So up until now, we have not been able to collect data since it takes a long time to get an experiment started up. And also we're trying to figure out the logistics of the experiment. But we have completed partially a pilot experiment where instead of the local injection of the cholinergic antagonist directly into the prefrontal cortex, we applied a systemic injection of cholinergic antagonist, specifically scopolamine, a muscarinic antagonist, to mice, and then measured their calcium dynamics in the parvalbumin interneurons in the prefrontal cortex. So I'm going to show the results, the behavioral data, as well as some of the photometry data from that experiment. So currently we have an n of 5, but as the experiment continues ongoing in the next couple of weeks, we hope to achieve an n of 8 and later start testing the nicotinic antagonist as well. So we find that in the saline condition, as well as a low dose scopolamine condition of 0.1 milligram per kilogram. Sorry, I'm just going to zoom in so you can easier see this. We see that in the low dose group, there's a significantly higher hit rate than in both of the high dose groups of scopolamine. So this is interesting because it actually confirms or agrees with an earlier study that also injected scopolamine and looked at sustained attention performance in a different task that didn't require the non-target stimuli. I mentioned it, that's included in the RCPT earlier. So in terms of false alarm rate, we see something similar, but just to a less of an effect. We see that in the low dose condition, there's slightly less of a false alarm rate compared to the high dose condition, which means animals were making less mistakes compared to when given the high dose of the antagonist. Similarly, in terms of discrimination sensitivity, which is essentially a score measuring how well an animal can differentiate between a target or S plus and S minus or non-target stimuli, we see that in the saline and low dose group, the mice was the discrimination sensitivity was significantly higher, so better able to discriminate compared to the higher dose scopolamine groups. So again, these are all in line with earlier studies, also doing systemic injections with scopolamine. So next, I want to show the photometry data that we've collected until now. So this is just an example trace of what we expect in from the photometry data from these studies. So you can see there's a continuous line, and these bumps and curves are indicative of calcium dynamics, so changes in calcium activity of these Parvalbumin interneurons in the prefrontal cortex. So currently, I have not calculated any statistical differences between the treatment conditions because we're still currently trying to figure out the best way to do so. So what I applied right now is the area under the curve, or AUC, during the 0.5 seconds before, 0.5 seconds after stimulus presentation. So in this region here, we chose to do this because we thought that would be the region where most of the changes in activity happen in these Parvalbumin interneurons during each trial. And we wanted to see, we wanted to have a quantitative measure to distinguish activity in Parvalbumin interneurons between the different treatment conditions. Over the next coming days, we're going to sort out exactly how we're going to test this significantly. This is the statistical test, the workflow through Python, and etc. But the statistical significance will be coming out soon, is what I'm trying to reiterate. So right now we can see a large degree of differences of trends in this data. We can definitely see in the correct rejection, we can see differences here. So again, in all of these data points, there's an N of 5. So hopefully as the N goes up, we can better see trends and test the statistical significance. But right now, I just want to show what I've collected so far. So moving on to the discussion. So as of right now, the behavioral data from our pilot experiment, so our scopolamine systemic injections combined with photometry data, is in line with behavioral data from other studies using scopolamine and measuring sustained attention. So we continue to work on our analysis pipeline for the photometry data, so we can test statistical significance, and we continue to build up our N numbers so we can better make sense of the trends seen in our data. Additionally, the two major experiments in my study, so the local injection of antagonists, as well as measuring of Parvalbumin interneuron calcium dynamics, as well as the optogenetics portion of my experiment, are ongoing. And I hope to have more data soon. Thank you very much.